Looking at cells, we're going to start by discussing the different types of cells and what a living system is. So this PowerPoint was put together with an outline that kind of explains the different aspects of understanding microbiology or cellular biology. So the first thing we want to talk about is defining life. So if biology is the study of life, then what classifies as living? These questions all are used together to determine whether or not something's alive. So if the answer is no to any of these questions, then it's not considered living. So for example, a table doesn't grow, it doesn't reproduce, it doesn't use energy. So a table is obviously not alive. Whereas a dog grows, reproduces, uses energy, responds to environmental stimuli, it has order to its body and systems, it regulates its internal environment, and it shows evidence of evolutionary adaptation. Therefore, it would be considered living. This led to the development of cell theory. Cell theory has three main parts. The first is that all living organisms are composed of cells, and there are some examples here for you. The second is that the smallest unit of life is a cell. So if you break open a cell and look at the parts of a cell, even though there are some subcellular structures in there that are smaller than cells, none of those can answer yes to all of the questions on the previous slide. And then the final tenet of cell theory is that all cells come from pre-existing cells and can't be made from non-living material. And so there was a famous experiment that was conducted in order to falsify or take credence away from a theory called spontaneous generation, and led to this development of all cells coming from pre-existing cells. And this was done by Louis Pasteur, a French scientist, who's better known for pasteurization. But an experiment that he did also is called the gooseneck flask experiment. And in this experiment, he took a flask that was open to the environment with an opening here and broth down in here, so think about like a soup. And he left it out on a counter for a period of time and determined that there were microorganisms that were growing on top of the broth by the time the experiment was over. However, he also had what's called a gooseneck flask because the top of it looks similar to that of a gooseneck. And this prevented dust particles from actually getting down into the broth. And on those dust particles, we know now, were microorganisms, things that you can't see with the naked eye. And because this experiment collected the dust down here, the microorganisms did not go down and inhabit the broth and live off the nutrients in the broth. So this was his idea, or his way of showing that cells don't just magically appear. They actually come from pre-existing cells that we can't really see with our naked eye. The size of a cell is determined by a relationship between the surface area and the volume of the cells. So we're going to talk about that in more detail. The surface area is responsible for exchange of nutrients in and out of the cell and waste products in and out of the cell, whereas the volume is responsible for cellular functions like respiration and others. When we look at this ratio, we're going to look at volume first, which determines the processing of nutrients and the development of waste inside of the cell. The metabolism requires import and export for it to work, so it relies on the surface area of the cell, which is responsible for importing and exporting substances from the cell. So in looking at the volume, we know that it relies on surface area, and the surface area refers to the membrane of the cell, that area surrounding the cell, separating it from its interior and exterior parts, separating the cell from its environment. So bigger cells will allow more substances in, more nutrients in, and more waste out, which means they can do metabolism at a faster or at a more successful rate. However, they have to be able to support that metabolism. So we want to make sure that there's an appropriate relationship between surface area and volume, because as a cell gets larger and larger and larger, the volume actually increases faster than the surface area. So we know that if we have the size of one length of the cell is 1, that a cube has 6 sides, so that would be, the surface area would be 6. and the volume is going to be 1. So the surface area to volume ratio is going to be 6. As you continue in this chart, when a cell gets larger, make the cell size 2, the length of a side 2, that means that you're going to have a surface area of 24, and that's because 2 times 2, which is 4, gives you the one surface, and then you have 6 of those surfaces surrounding the cell, so 4 times 6 gives you 24. And the volume is going to be the cube of one of the sides, so that's length times width times height. So we've got the volume is 2 times 2 times 2, which is 8. The ratio, the surface area divided by volume, would be 24 divided by 8, which makes it 3. As you get larger, a cell with a side of 3 would give you a surface area of 54, a volume of 27, and a surface area to volume ratio of 2. As you continue getting larger and larger and larger, the surface area to volume ratio continually gets smaller and smaller because the volume increases at a faster rate than the surface area. As you can see here, volume is in pink, and it increases at a faster rate than surface area. So if we were going to compare this ratio, surface area divided by volume, you would see that as the size of the cell gets larger and larger, this ratio gets smaller and smaller. So why is that a problem? Well, the problem is that bigger cells have more metabolism, but they have a harder time getting substances in and out of the cells. So there's going to be some limit to the size of cells. And one way that organisms get around this limit is to become multicellular. So you can put extensions, like inside your intestine, you have these microvilli, which help absorb nutrients from your digestive system. Those are simply one cell, and they're just arranged in a manner that maximizes the surface area so that the cells themselves don't have to be large. This is not just one big cell. This is called the brush border because it's composed of a bunch of small cells that allow for transport across their membranes. Another way is to flatten out, and so you've got some cells that get very, very flat so that you get a greater surface area to allow the transport of nutrients and waste products. You also divide to become multicellular. So here we have a zygote, which is the fusion of an egg and sperm cell, and then it divides to form an 8-cell embryo and continues to divide to form a fetus. And so we've got multicellular organisms like flowers that can show examples of this with root systems underground composed of many, many cells. So because you're limited in the size that a cell can grow, you're going to divide that cell and become multicellular, many cells that will all operate as one organism. We've also got human examples of this, and again, this is a slide showing the intestines of a human, which are adapted to maximize surface area so you can absorb the nutrients that you put into your system. There's two main categories of cells. There's prokaryotic cells and eukaryotic cells. When you think prokaryotic cells, think bacteria, because mostly this is composed of bacteria. When you think eukaryotic cells, think everything else, including humans. So we have a nucleus inside of our cells, which contains and protects our DNA. Our DNA is linear in nature, meaning that it can be arranged in chromosomes that can be moved during cell reproduction. We have histone proteins that are involved in the formation of our DNA and help us supercoil our DNA into chromosomes so that they can be moved around when necessary. We have organelles that are membrane bound, and we have complex ribosomes, whereas prokaryotic cells don't have those features. These are two big categories of cells. Think bacteria, no nucleus, circular DNA, no proteins associated with their DNA. All of their organelles consist of non-membrane bound, which means that we have an endomembrane system. We have ribosomes that are free-floating, and we have ribosomes that are attached to the endoplasmic reticulum, which you'll learn more about in this unit. Our next category, and these are both types of eukaryotic cells, they are animal cells and plant cells. And there's if you Google image any of this, for instance, animals, if you go Google animal cells and plant cells or prokaryotic cells and eukaryotic cells, you'll see some really fantastic drawings of this, and they're well labeled. So take a look at those. I'm going to highlight some of the differences between plants and animal cells. In animal cells, but not plant cells, you have lysosomes, centrioles, and flagella. So a lysosome is used in the breakdown of macromolecules because we ingest these large things like proteins and carbohydrates. Our lysosomes help break those things down. Centrioles are used in our cell reproduction. We'll talk more about that in mitosis and meiosis. And we have flagella, which help the cells to actually move. In the case of the best example I can think of a flagellum would be a sperm cell with its tail. The tail is a flagellum, which allows it to move so that it can carry out reproduction. In plant cells, but not animal cells, there's a cell wall to give it a rigid structure so it can stand up and not move very much. The centrosomes are used instead of centrioles during reproduction. More on that later. Chloroplasts, which are used for photosynthesis, and there's a central vacuole, which is this big water-filled container here that helps it maintain its structure. This is a website I think is worth your time to go visit. It brings up a list of the organelles that are inside of cells and what each organelle does. So that is important for us to know, but I'm not going to go into detail on that now. We can talk more about that in class and one-on-one. The last thing I want to make sure we understand about cells, because we're talking about things that are too small to be seen just with your eyes, is what magnification means. Magnification is the size of the image divided by the actual size of the specimen. And in order to do magnification calculations, you've got to make sure the units are the same. So in this example here, which comes from an old IB biology exam, they're saying that the actual diameter is 8 micrometers, but the student has drawn it with 40 millimeters, the diameter being 40 millimeters. So what's the magnification of the drawing? And we'll need to go through this practice problem. I want you to attempt it. You've got to convert to the same units and then put the size of the image divided by the actual size to get the correct answer. So feel free to talk to me about that and what you think the right answer is.